Welcome, everybody. My name is Sarah Cohen, and I'm the administrator for the Center for Parkinson's Disease at Stony Brook Southampton Hospital. Thank you for joining us today for the Parkinson's Disease Awareness Month lecture series, a collaboration between Stony Brook Medicine, NYIT College of Osteopathic Medicine, the Friedberg JCC Oceanside, and in partnership with the American Parkinson's Disease Association. We will be meeting Fridays in April via Zoom, offering a series of lectures and Parkinson's-specific programming that celebrates the Parkinson's community here on Long Island. I do hope that you will join us each week. Today's webinar discusses the COVID-19 vaccines as well as issues related specifically to COVID-19 and Parkinson's. We have two presenters today, Dr. Fredric Weinbaum, Chief Medical Officer and Chief Operating Officer, Stony Brook Southampton Hospital, and Dr. Hanna Czarkowska, Movement Disorders Neurologist at Stony Brook Parkinson's and Movement Disorders Center. There will be designated time at the end of the lectures for your questions and answers. I encourage all of you to type your questions as they arise in the Q&A box located at the bottom of your Zoom screen. And now I am thrilled to introduce you to Dr. Fredric Weinbaum, who will be discussing the COVID-19 vaccines and how they impact us in our daily life. Dr. Weinbaum, I hand it over to you. Thank you.
Thank you, Sarah. And first off, I want to thank Sarah because she's a well-recognized natural resource at Stony Brook Southampton Hospital, and worth her weight in gold as any natural resource is. So thank you, Sarah. You're doing a terrific job for us and for the community and for all the people with Parkinson's that we serve here on the East End. I've been asked to discuss a little bit about the vaccines that are currently available to treat or to help prevent SARS-CoV-2 or COVID-19. Let me start by saying that SARS first arose around the turn of the century, around 2002. The first cases of SARS were discovered, and that was a variant called CoV-1, SARS-CoV-1. SARS stands for Severe Acute Respiratory Syndrome. It was rapidly determined that the disease was caused by a zoonotic virus, which means it's a virus that arose from another species and ultimately was transmitted to man, and it arose in China and actually infected slightly under 10,000 people and had a case fatality rate of 10%. So it was a very serious illness, but was able to be controlled and never became a pandemic. So we knew it was out there, and even at that time, it was determined that the spike protein, which is that part of the virus which attaches to target cells and enables the virus to enter into the cell and begin replicating, was the presumptive target for a potential vaccine. Now, the spike protein is an interesting protein in that it's capable of several changes in conformation. So in order to produce a vaccine, it had to be stabilized in something called the pre-perfusion state, so that antibodies and immunity could develop to the molecule that was the spike protein that sits on the virus surface before it fuses with the cells that are ultimately the target in the human body. So the first vaccines that were developed when this current episode of SARS-CoV-2 arose were developed using a very exquisite and very beautiful technology using messenger RNA. So let me start by saying that SARS-CoV-2, the cause of COVID-19 disease, arose towards the end of 2019, and it was presumed to be another zoonotic infection, just like the original SARS, although the actual site and place where it arose and the source of the virus remains to be fully scientifically determined. But the virus, we know, arose in China, and we assume, like the original, that it probably came from the bats, but we'd like to see that proof. There are three current vaccines that have received emergency use authorization. All three vaccines are aimed at producing immunity to the spike protein, the protein S, in its pre-fusion state, which means as it exists on the native virus prior to fusion with cells. And the exquisite science of it is that if you can produce an antibody response that attacks that protein, you will in fact prevent the virus from entering cells. And if it does enter cells, you will also produce the appropriate cellular immune response through T cells to attack the cells that are infected and destroy them as well. Now, there are two vaccines that are based on messenger RNA. Regardless of what anyone has told you, messenger RNA does not do anything to your DNA. It doesn't change who you are as a person. It doesn't do anything that is theorized in various conspiracy websites. What messenger RNA is, it's simply instructions to your cellular mitochondria, which are the machinery that produces protein, to have some of your cells produce spike proteins. And those spike proteins then trigger an immune response. The messenger RNA, though, is in and of itself rapidly destroyed, both in the serum and intracellularly after it's processed through the mitochondria. So in order to deliver it into cells, it has to have a specific envelope that is been manufactured by both Moderna and Pfizer, have different methods of producing this stabilized lipid envelope that can deliver the messenger RNA into some of your cells, which then develop the machinery to produce spike protein through their own mitochondria and display that spike protein on their surfaces and trigger the immune response through the antigen detecting cells that circulate in your body. So the Pfizer vaccine and the Moderna vaccine are both based upon this messenger RNA technology. They differ slightly. The Pfizer vaccine requires a much more stringent cold chain than the Moderna vaccine. By the cold chain, I mean it has to be kept at a super cold temperature to be stored for long durations. But now, through time, we've learned that even the Pfizer vaccine can be kept in a freezer at regular temperatures for a month. So the differences between the two are narrowing, but have not narrowed entirely. The Moderna vaccine requires 2 injections 4 weeks apart, and the Pfizer vaccine requires 2 injections 3 weeks apart. The third vaccine that's been approved is a Johnson & Johnson vaccine that's based on a different method. The Johnson & Johnson vaccine is based on a human adenovirus type 26, that has been rendered replication incompetent. I mean, it's a DNA virus that has been genetically altered so it cannot replicate. Those genes that are required for replication have been removed through genetic manipulation. So that virus is grown in a tissue culture of something called the per.C6 cell line. And that adenovirus can't replicate or cause disease, but it has been genetically given the capacity to produce spike protein. So in essence, you're getting a adenovirus, local adenovirus infection. It can't cause a true human infection, but it can enter into cells. The DNA from that virus produces messenger RNA that instructs your mitochondria to produce spike protein. And that spike protein is presented to antigen detecting cells, and then to human T cells and B cells, helper T and B cells, and antibody producing cells. And a potent immune response to the spike protein is produced. Now, all three vaccines have received something called an emergency use approval. That approval requires presentation of a large body of data to a blue ribbon panel of experts who deem the vaccine to be both efficacious and safe to administer. The Pfizer vaccine trial had 43,500 people within it. The Moderna, 34,500. The Johnson & Johnson, 43,800. In all three trials, there were no fatalities that could be ascribed to administration of the vaccine. All three vaccines do produce some side effects, local pain and tenderness, some potential for low-grade or even higher fever, some potential for muscle aches. All of the symptoms from the vaccine, in essence, are amenable to symptomatic treatment and disappear within 48 hours. So the vaccine is safe. The incidence of anaphylaxis, which is a severe complication of vaccine, is around 5 per million. And to date, there are no reported deaths from anaphylaxis, because when it's given, everyone observed, and after the observation period, anyone who does develop any signs of anaphylaxis goes to hospital. Treatment occurs, and everyone is recovered uneventfully. That's been reported to the CDC and in our literature. So the vaccine is extraordinarily safe. It's one of the safest vaccines that is around there. There are some side effects, but they're all tolerable. And all the vaccines produce very high levels of immunity to all the strains that are known and out there. And by that, I mean the Moderna vaccine and the Pfizer vaccine, 2 weeks after the second dose, was effective in preventing clinical disease in approximately 95% of the cases in both trials. And the Johnson & Johnson presented clinical disease 2 weeks to 28 days after administration in about 70 to 75%. However, after 49 days with Johnson & Johnson and 2 weeks after the second dose with Moderna, and a little bit more after the second dose with Pfizer, none of the patients required hospitalization for serious Pfizer serious coronavirus infection. So just to reassure you, if you want to think, when am I really immune after you get that first shot? I would give yourself 6 to 7 weeks to really feel that you're at maximum immunity from any of the vaccinations that you are receiving. And although the neutralizing antibodies being produced are less effective against one of the strains out there, the South African strain, that's not by an order of magnitude. It's about 2/3 less effective, and it's believed that that's adequate for prevention of serious coronavirus infection. Now, once you get immunized, it does not mean that you give up doing your preventive measures. It is possible to get an infection after immunization, although the infection will extremely likely be either asymptomatic or have very mild symptoms. But in order to protect others, we do recommend you continue to wear your mask in communal situations where you could encounter unvaccinated individuals, and continue to do social distancing as you have are currently doing now. There are other vaccines that are in the offing that are based on slightly different technology. Now, one thing that's out there with the Johnson & Johnson vaccine, the cell line, the per.C6 cell line, was originally produced from embryonic retinal cells derived from an 8-week-old aborted fetus in 1985. It's been determined by papal edict. First of all, that's hundreds of thousands of generations away from that cell line. And it's been determined by papal edict that none of the cell line produced vaccines should be considered to be in any way sinful. So if anyone is resisting Johnson & Johnson because of religious reasons, the Catholic Church has determined that it's not immoral to be taking a Johnson & Johnson vaccine. But it's out there, it's on the internet. People make a big deal of it. It's hundreds of thousands of generations removed. It is perfectly safe, and it's an excellent vaccine. And 7 weeks after taking that vaccine, 100% of patients in the trials were prevented from having hospitalization and serious infection due to COVID-19. Now, I really think that the race is on. We really want to get as many people vaccinated as possible so that we can get a sufficient number of people vaccinated to have herd immunity and to be able to finally put an end to this pandemic. There's another vaccine that's being given in other countries, the AstraZeneca Oxford vaccine. There's a significant supply of that vaccine that we have in stockpile, but that has not received emergency use approval here. There are a number of issues with its trials. They are attempting to redo some of it and to be able to present that with emergency use authorization. There have been some reported cases of thrombosis, and so there are some questions about its utilization. But that is not one of the vaccines that is available by emergency use authorization in this country. So vaccines are safe, they're highly efficacious, they're moral, and everyone should take it. There should be no inherent hesitancy about any of these vaccines. They're really exquisitely developed. They're beautiful technology and highly effective. And it's really that beautiful new up-to-date technology that enabled them to be developed so rapidly. One other note, and that's beyond vaccines. There are currently in development, and one's about to enter phase 3 trials, some fairly effective therapeutics we hope that may serve as really excellent things to do for patients if they are to be infected. And there are in the works boosters. And finally, for children, there's a current trial with the Pfizer vaccine that we hope will prove effective in children. We do know it does produce a very strong immune response in children. And finally, for those of you who are pregnant or contemplating pregnancy for your children, for your grandchildren, the American College of Obstetrics and Gynecology has come out and recommended vaccination. And we think that there's no theoretic reason why it would not be safe, although it has not been fully tried and trialed in pregnant women. We think theoretically there's no reason to believe it would present any undue risk. We do know for sure that we are seeing pregnant people with COVID-19, and that does present a significant risk. Now, I believe my 20 minutes are over. I'd like to turn this back to Sarah to continue. And questions will be entertained at the end.
Dr. Weinbaum, thank you so much. That was incredibly interesting, and I learned a lot, and I'm sure all of our attendees have as well. And as Dr. Weinbaum mentioned, we will have a Q&A at the end of the webinar. So as your questions come up, please do put them in the Q&A box at the bottom of your Zoom screen, and we will make sure to get to them at the end. Now I'm thrilled to introduce Dr. Hanna Czarkowska, Movement Disorders Neurologist at Stony Brook Parkinson's and Movement Disorders Center, who will be speaking about COVID-19 as it relates more specifically to Parkinson's disease. Dr. Czarkowska, take it away.
Thank you, Sarah. Thank you, Erin, for your great initiative. Good morning, everybody. Pleasure and honor to be here. I'm going to spend the next 15 minutes or so discussing what we have learned over the past several months with regards to COVID-19 and Parkinson's disease, how this infection affects the population of Parkinson's disease patients. I have a few slides actually that I wanted to share to keep myself on track. I have some trouble pulling them up, but they're not necessary actually. So, you know, I would like to answer those questions that we were asking ourselves as neurologists and movement disorders neurologists in particular back in the spring of 2020, and we really didn't have any answers for them. One of the most common questions I was being asked back then was about risk of infection itself. Am I, as a Parkinson patient, more likely to catch this new virus or not? And the results of studies, as we call them, epidemiological studies, so studies specifically looking at the risk of a problem, in this case, a risk of infection in specific populations, started coming out in publications around June, July of 2020. Long story short, based on them, and they differ in their size, in their accuracy, in what specific populations they were looking at. Some of them were based on pre-existing registries of people with different backgrounds, different medical conditions. Some were more telephone interviews. The message they conveyed to us was that Parkinson's disease in itself does not seem to be a risk factor for the infection. So very good news, actually. it's not like diabetes or dementia or smoking that definitely puts you at a higher risk of getting the virus, of becoming positive. Parkinson's disease does not seem to be one of them. In fact, just as a curiosity item here, there were some reports and some interesting theoretical speculations and explanations behind those reports that were trying to make a point that Parkinson's may actually prevent you from getting infected. I wouldn't get too excited about them. They were small sized and they weren't enough to feel overly confident. But at least what we seem to know is that Parkinson's disease does not carry any increased risk of getting the infection itself. The next question was with regards to COVID-19 symptoms. Will they, if I do get the infection, if I do get COVID, will my symptoms be any different than if I didn't have Parkinson's in the first place? And just like Dr. Weinbaum said, SARS stands for the severe acute respiratory syndrome, but we learned very early on that it's much more than just a respiratory illness, that there are multiple systemic symptoms, such as fever, such as excessive fatigue and headaches, and the infamous loss of smell and taste, and that the high percentage of people with the virus had stomach upset or nausea or diarrhea. So the variety of symptoms is out there to begin with. It has been reported in Parkinson's patients that sometimes the only manifestation of the disease is sudden worsening of their Parkinsonism. So if you wake up one day and you're much stiffer than you used to be, and you're tremoring much more, and you can't walk, and it feels like your Parkinson's all of a sudden took a nosedive, and it's so much worse, you have to suspect something else is going on. And we've known that for years. And we always convey to patients that it's not a normal trajectory of Parkinson's progression, that typically there is something else up. And this something is frequently an infection. And it may be, in this kind of new world we live in, it may be COVID as well. So it wouldn't be unnecessarily unreasonable if you're feeling your symptoms, your Parkinson's symptoms much worse all of a sudden, to get testing for COVID on your list of things to do. With regards to anti-Parkinsonian medications, there are questions out there. What should I do about my medications if I do get sick? Should I change anything about what I'm doing? Increase the dose, reduce the dose, start taking something new, avoid certain medications? There's nothing to avoid that we know of as far as anti-Parkinsonian medications, because they does not seem to be making COVID-19 worse. They don't seem to be interacting with any of the treatments out there that are used for COVID. So definitely nothing to stop. As I said, you may experience some increase in your Parkinson's symptoms, so it may not be unreasonable. And it has been, again, proven and documented in large sized studies that many people with Parkinson's who got COVID needed to transiently increase the doses of these medications. So that's perfectly reasonable. And again, as a side interesting note, some of the anti-Parkinsonian medications have been postulated to have some protective effect. It's more of an academic interest at this point. We do not recommend starting any new medications, but not unexpectedly, I think one of them was amantadine. You may know that amantadine has been developed as an anti-flu medication to begin with, so it has some efficacy against viral infections. And there were what we call case series, so series of patients reported in literature who were on this medication on amantadine to begin with, either for Parkinson's or for some other indications. And they were all exposed to the virus, and they remained asymptomatic. again, in larger scale studies, that hasn't been proven that the medication, change the course or outcome of COVID-19, but it's something, interesting that may need kind of further look into. Next question, is my Parkinson's going to get worse or get worse faster if I have COVID? What are the short-term and long-term consequences on my Parkinson's if I do have COVID? So in a short run, like I said, you definitely can experience that, something's worse, that if you're feeling under the weather, for whatever reason, your Parkinson's may feel worse temporarily. Is it going to change your future in a way? Is it going to make the progression any faster? We don't know. There is just not enough time that has passed to have any confidence in answering this question. There are, again, theoretical reasons to believe that actually this infection, like many other viral infection, may make people more prone to develop Parkinson's to begin with. So people who are now, healthy and young and have no risk factors, as we know, may be at a higher risk of Parkinson's if they have COVID now. Is it going to pertain to people with existing disease now and the risk of their faster and more rapid progression in the future? We have no idea. I'm sure, as we move along, months and years from now, really, we will have some data on that. And the last question, probably, most difficult and most feared in a way, is my Parkinson's putting me at a higher risk of, we know about infection itself, but higher risk of a bad outcome, of a bad course and a bad outcome? Am I, because of the fact that I have Parkinson's, more likely to end up in a hospital, to end up with a tube, or to die, right? So with regards to the last, the numbers are all over the place, really, from the studies. So there are some in which mortality among Parkinson's patients with COVID was as high as 75%. And then at the same time, there were others talking about 40% and then 20% and then below. There were some groups of patients who had all odds stacked against them. They were kind of deep into their disease. They were in nursing homes. They had other medical problems, and they fared really well. They would, again, either stay asymptomatic or had only mild symptoms. They may or may not needed oxygen, but would not end up in the ICU level of care, and they weren't dying. So what seems to be increasing the risk of a bad outcome is later stage of Parkinson's and longer duration of the disease, and also dementia that some people, develop over the course of their disease. These things seem to be making you more likely to have a worse outcome than not, but there's a huge variability out there. So kind of sum it all up, and I will attempt to share my screen one more time, and now it's successful, looks like. Let's do it. All right. Here. Okay. So once again, Parkinson's disease by itself does not seem to predispose to the infection, although those data are coming from very specific areas in the world. A lot of them from Italy, very little from China, some from the United Kingdom, some from Spain, some local here from the US. We don't have a worldwide data available on the risk of infection. As I said, mortality may be somewhat higher than in general population, but the variability is huge, is enormous. The risk factors of worse outcome probably include later stage of Parkinson's, longer disease duration, dementia, and prior strokes. And with regards to anti-Parkinsonian medications, we do not have enough evidence that they impact in any way a course or outcome of COVID-19. And that's pretty much all I have to say. I'll be happy to take any questions. And thank you again for tuning in.
Thank you, Dr. Czarkowska, so much. That was also incredibly informative and interesting. So thank you for that presentation. I do open it up to questions and answers. We'll go ahead and get started. Oh, sorry, my video is not on. Let me turn it on. There we go. If you have questions, go ahead and type them in the Q&A, and I will continue to keep an eye on it and ask them as we are able. The first question I'm going to direct to Dr. Weinbaum, and I'm going to bundle a couple of questions if you don't mind, is if I've had a COVID-19 infection, should I still get vaccinated? And if I have been vaccinated, do I require a booster at some point?
Okay. Question one is one where it can be pretty definitive. And yes, you should be vaccinated if you have had an infection. The truth is that the degree of immune response with neutralizing antibodies is actually better after vaccination than after native infection, particularly with a mildly symptomatic or asymptomatic native infection. The other thing that's really now being reported is that those people who are vaccinated after having had an infection in the past do develop fairly robust immune responses to the vaccine, and they do have high levels of neutralizing antibodies. So it's a definite positive to be vaccinated. In terms of, I see a question out there about the effectiveness of vaccine. I did mention that the South African variant, it's the B.1.351 variant. That variant has some amino acid replacements in the protein that render it less susceptible to neutralizing antibodies. And that statistic of a lower susceptibility means that in the test tube, the amount of dilutions of the serum that you can do before you neutralize an aliquot of virus and put it in a culture is reduced, so that you can do many more dilutions and neutralize the 1.1.7 variant from the United Kingdom, the one that is predominant here in the United States now. That's neutralized to a greater extent by serum from patients who've been immunized. That doesn't mean we have no immunity to the South African variant, and it's not an order of magnitude. So usually, in order to take you to a point where you might not have sufficient neutralizing antibodies, it needs to be an order of magnitude. Now, so we still believe that the immunity to the vaccine will be good for the South African variant when it's fully developed. There may be some variants that arise in the future that do require booster shots. So that question of will we need boosters, the answer is it depends upon what variants arise, number one. And number two, it depends upon the durability of the immune response. And the answer is not yet out as to whether there'll have to be an annual type of booster shot. Our hope is no, but we don't know for sure. And finally, as therapeutics come out, the whole requirements may change.
Thank you very much. I have a question for Dr. Czarkowska. Is Lewy body dementia at a higher risk for COVID-19 infection?
So to my knowledge, there is really one study out there coming from, I think it's from Bologna, that tried to separate Parkinson's disease and other forms of Parkinsonism, not only Lewy body, but other forms of Parkinsonism as well. And one thing that was evident in this study was that people with atypical Parkinsonism were at a higher risk of hospital admission. There was really no difference in terms of, mortality and bad outcome, but they were more likely to get admitted to the hospital. There's a lot of evidence behind dementia in general, and as we know, Lewy body disease, is really a dementing illness, and dementia is the main feature of Lewy body. People with dementia of any sort are definitely a higher risk of both infection and a bad outcome. So there is a very strong association, not specifically for Lewy body, but for dementia in general.
Thank you. Dr. Weinbaum, a couple of sort of logistical questions, which I'll send your way if I may, and that is, now that I am fully vaccinated, what is it safe to do? So is it safe to travel? Can I visit my grandchildren? If you could speak briefly on that, please.
Okay. Well, first, with travel, airplane travel is felt to be very safe for a vaccinated individual. Of course, you're wearing a mask when you do it, and you're maintaining social distance in the process as best you can through it. But airplane travel is believed to be safe, and that's what the CDC has said. Can you visit your grandchildren? The answer is that what's felt to be true is that you can have small group gatherings and unmasked situations with other vaccinated individuals. Children are at very low risk, but it has to be an individualized decision. If you're vaccinated, you have a very, very low likelihood of having a transmittable infection, but it's not totally excluded. I think many people who are vaccinated are taking that small risk, but I personally think it's okay. But once more, you want to be in small groups, not large groups, and if possible, all of the adults you come into contact with should be vaccinated. Before too long, I hope that the trials come through and we're able to vaccinate children as well. The younger the child, the lower the risk of disease. There are very rare cases of severe illness in young children.
Thank you. There have been a couple of questions come through, which I'll put out to both of you actually as it overlaps. Is there any evidence to suggest that the COVID vaccine itself can worsen Parkinson's symptoms? And if it does, are those changes enduring? Perhaps Dr. Czarkowska, do you know?
Sure. So I personally haven't seen any reports on that. I am going to assume that, just like I mentioned, there is expected to be transient worsening of Parkinson's symptoms if you get the disease, right? Or if you get any kind of infection or any kind of issue to your immune system that's occurring. So it would not be unreasonable to think that that there may be a transient increase in your Parkinson's symptoms after the vaccination as well. But I don't think, and I'm not aware of any specific studies that look into that.
Thank you. Dr. Weinbaum, a couple of questions about the logistics of developing the vaccines. One question asked, I'm going to put two together here. In clinical tests, are people actually given COVID is the first question. And are there other companies also working on vaccine development?
Okay, the first question is no, it's not considered to be ethical to give someone a disease as part of a clinical trial currently. Whether that ever occurs in the future, I don't know. But for now, all the trials are done with people who are living in an area with a high prevalence of disease, so that the numbers required in order to get statistically significant results are high, which is why you need, close to 35 to 50,000 people in a trial with somewhere between 20 and 25,000 people, in each arm of the trial, to see that some people in the trial will get infections. So, for instance, in the Pfizer trial, there were 43,500. Half of them got the vaccine, half didn't. 2 weeks after the second dose, if there were, 100 people that got infected in that trial, 95 were in the group that got the control, the vaccine without the spike protein mRNA, and 5 were in the group that had the actual real vaccine. So you can say it's 95% efficacious. That means that 95 people were in the control group and 5 people were in the group that got the vaccine. So you would have expected similar numbers of infected people in both arms, but instead it was 95% in the control group. So that's what's meant by efficacy, and that's how it's measured, and those are how the trials are done. The company that's now producing a vaccine that is based on a spike protein that's has something called an adjuvant, which increases the number of antigen presenting cells that come into an area of vaccination. That is a company called Novavax, and they actually have isolated spike protein. They grow the spike protein in a tissue culture line, and then they isolate the spike protein, combine it with an adjuvant that's given as an injection. The adjuvant helps draw in more antigen presenting cells, and you get immunity, high level of immune response to the spike protein, which is manufactured artificially in culture. The Novavax uses to grow their spike protein, they use something called the baculovirus, with a modified spike protein in tissue culture of non-human virus, non-human cells. So it's a entirely different technology.
Thank you. Another vaccine follow-up question. After I've been vaccinated, can I still carry and infect other people?
And the answer is we're learning more and more as we go along. But the answer is yes, but a very, very low probability. So now they've done some testing with Johnson & Johnson vaccine more than 7 weeks after the initial immunization, looking for asymptomatic infections, and they haven't been finding any asymptomatic infections, or very, very, very few. So the chances of having an asymptomatic infection, thereby infecting others, is not zero. What we believe it's very low, and because it's not zero, we want you to wear a mask. Obviously, if you develop symptoms, it's possible, as you say, if it's 95% efficacious, it means 5 of the people who got the infection out of 100 who got the infection were in the vaccinated group. They all had mild infections. None of them ended up in hospital in the trials for Moderna or Pfizer, but they presumably had a 2 to 3 day period where they could have transmitted it to others.
Thank you. A couple of 2 other vaccine questions. The first one is there a difference in size of dosage for adults and the proposed children's vaccine that is being researched?
I don't know. I haven't seen the trial published, but the answer is likely yes. The way they measure the Moderna and Pfizer is in micrograms, and the number of micrograms is likely to be different for children than it is for adults. Those trials are currently being performed. The initial trial is one where they give varying numbers of micrograms to children. Then they show the immune response, and it looks like the immune response in children is very pronounced at a fairly low dose of the Pfizer vaccine. So my hope is that within a year we'll have a vaccine for children.
That's great. Thank you. And then the last question, there have been rumors circulating that you shouldn't take Advil after the vaccine, or you shouldn't take Tylenol, or you should take Benadryl before having the vaccine. Can you speak to that, Dr. Weinbaum?
Well, the first off is, the recommendation is not to take any of that before the vaccine, but that recommendation is not based on any clinical data. That's just based on some theoretic considerations. That recommendation may not actually be true. In terms of post-vaccination, everyone who is an authority believes that if you have symptoms, to treat yourself symptomatically with Tylenol or Advil or Benadryl, if you get a rash, certainly would be fine. And there's no evidence that there's any harm. There are no clinical trials that address it. There's no theoretical reason not to do it once the antigen presentation has occurred.
Thank you. Someone writes in, would you please explain what is an asymptomatic infection?
Well, what an asymptomatic infection is where the virus gets into your cells in your respiratory tree, that you develop an infection, but the number of cells involved are not sufficient to make you feel sick. And the disease may be short-lasting, and before you develop a sufficient burden of virus and or infected cells, the infection is eliminated. And that has occurred. We know there are those type of infections do occur because, people who are contacts have been tested, they test positive, they have no symptoms. 3 days later, they test negative. They had an infection, they've cleared it. The way the virus works is it depends upon the number of copies of virus that you are exposed to. The larger the number of copies, the more likely it is you'll develop a clinical infection. And if you have a clinical infection, the more likely it is that you'll transmit it. Within any clinical infection, there's a period of time where you're pre-symptomatic, where you're shedding virus, but you haven't yet developed any fever, muscle aches, cough, etc. But you are shedding virus. So there's pre-symptomatic, asymptomatic, and that's, a major concern. And that's why this has been such a difficult to control pandemic, because of that pre-symptomatic and asymptomatic spread. If everyone who had the infection immediately got a fever and muscle aches and isolated themselves, the pandemic would be over.
And final question, in speaking about traveling, how safe are hotel rooms?
The one thing that is now pretty much agreed on is that contact spread from inanimate objects is exceedingly rare, and that it's in a hotel room that's been cleaned with ordinary cleansers is going to be safe. Ordinary cleansers are probably sufficient to clean and decontaminate any infected sites. And there are very few cases. Just about every case that has been studied has a known contact with someone who was shedding virus.
Well, thank you all so much for attending. I would like to extend a heartfelt thank you to Dr. Weinbaum and Dr. Czarkowska for your time and expertise this morning. I learned a lot, and I am sure all of our attendees did also. This webinar was recorded. I'm happy to share that. And I will say,
Sarah, I learned a lot from Hanna.
Yes.
Thank you.
Vice versa.
I hope everybody will join us for the next 3 Fridays. If people have questions, my email is on the Zoom registration confirmation. You can reach out to me directly, and I will say goodbye. Thank you all so much for being here.
And, thank you, Sarah, for everything you're doing.
My pleasure. Be well, everyone.
Stay well.
Bye-bye.